Okay, welcome everybody. We should be live now. Let me see. I'm Gillian Peterson. Welcome to today's MicroSeminar. I'm very excited to present our guest, Christina Faherty. I hope I've pronounced that right. We didn't get a chance in in our preparations to ask questions like that. Yeah, it's great to have you here today. She is an assistant professor at Harvard Medical School and Massachusetts General Hospital. And she started out her undergrad studies in biotechnology at Rutgers University. She comes from New Jersey, yes. A little bit south of Boston, yes. Where it's a little bit warmer, I hear. Yes. And after that, moved on to a PhD in infectious diseases at the Uniformed Services University of the Health Sciences. She got her PhD in the same year as I did, 2009. It was a great year. And after that, moved on to positions as a research fellow and a postdoctoral fellow in bacterial pathogenesis with PIs James Nataro and Eileen Barry at the University of Maryland School of Medicine. In 2013, she was called as a lecturer to Harvard Medical School and is an assistant professor there since 2014. Yes. Today, she's going to talk to us about her favorite microbe, I assume, or is there a microbe you love more than Shigella?
No.
Yeah. We're really excited to hear your talk. Thank you so much for coming today. If you're listening in live, you have the opportunity to answer questions in the YouTube live chat. Put in your questions at any time now throughout the talk towards the end, and I'll pose these questions to Christina at the end of the seminar. So take it away, Great. Christina.
Thank you very much. I'm going to share my screen here real quick. So is that good?
Looks good to me.
Okay, great. I'm waiting on there we go. Thank you very much for the opportunity to present here today. I'm very excited to interact with such a broad audience. So I'm very grateful for this opportunity. And I have actually been working with Shigella since graduate school. So I would by far say that it is definitely my favorite pathogen. So today we're going to look at how host factors affect Shigella pathogenesis. And so I first like to start off with a good note that Shigella, like other pathogens like E. coli and Salmonella, really contribute to the global burden of diarrheal disease. And luckily, in the past 20 years, we've made significant progress in that fight. And that's mostly due to clean water, sanitation, better nutrition, and the rotavirus vaccine that has really decreased infections and deaths. However, we still have a long way to go. At least pre-COVID, diarrheal disease was the 9th leading cause of death around the globe, and it especially affected children in developing countries under the age of 5 years. So especially for bacterial pathogens, like the three I've mentioned, current challenges include malnutrition, antibiotic resistance, and a lack of effective vaccines. And despite really researching these pathogens for the past 40 years and gaining significant knowledge about disease mechanisms and virulence, we still unfortunately have not been able to develop robust, effective bacterial vaccines. So new insights and approaches are urgently needed to help overcome these challenges. So the main pathogen I focus on is Shigella. And in case anyone's not familiar with this bacteria, it's a gram-negative pathogen. It is human and adapted, and that's important to remember. So it's really co-evolved with humans and primarily affects humans and higher primates. So there's really no adequate animal models that really recapitulate human infection well. And that's been one of the challenges that we've had with studying Shigella. So there are 4 species and multiple serotypes. The 2 predominant species are Shigella flexneri that affect developing countries in Africa and Southeast Asia for the most part, as well as Shigella sonnei, which tends to infect more industrialized nations like in Europe and in North America. There's also Shigella dysenteriae and Shigella boydii. For the most part, all the Shigella species are very similar and homologous, but there are some key differences. For example, Shigella sonnei encodes a type 6 secretion system that is important for pathogenesis, and that does not occur for Shigella flexneri. So these pathogens cause watery diarrhea or bacillary dysentery. And in order to do that, they basically secrete enterotoxins, which contribute to the watery phase of diarrhea. And then through invasion of the colonic epithelium is where you get the bloody diarrhea and the tissue destruction that's known clinically for Shigella. So I would like to note, I mostly focus on Shigella flexneri, and despite it being mostly prevalent in developing nations, it does occur in industrialized nations as well. And there is a current outbreak in Vancouver, Canada, that is affecting the homeless population. So in order to cause infection, the bacteria invade the colonic epithelium. They are unable to invade from the apical surface and must find these specialized antigen sampling cells known as M cells. Now, these M cells are actually few and far between in the colon, especially relative to the small intestine, which is interesting as to one of the interesting questions as to why Shigella prefers to infect in the colon versus the end of the small intestine. But anyway, the bacteria is able to traverse these M cells to gain access to the basolateral pole. They first need to evade innate immune cells like macrophages, and they do so by invading the cells where they're able to recognize that they are in fact inside these immune cells and very quickly induce cell death. Once they're able to bypass the immune cells, they're able to invade the epithelium at the basolateral pole, where they can then replicate and spread from cell to cell inside without having to re-enter the extracellular environment. So some things that Shigella does to help facilitate infection is regulate neutrophil migration to the site of infection. So early in infection, the bacteria likes to induce interleukin-8 secretion from the infected epithelial cells, which helps attract neutrophils to the site of infection. And that process, as they begin to disrupt the tight junctions, allow the bacteria to gain better access to the basolateral pole for invasion. But then once they get later in infection, the bacteria try to suppress IL-8 and the neutrophil response, since it's the neutrophils that actually destroy the epithelium and control infection and account for the bloody phenotype that is seen with diarrhea. So two more pieces of information that are important to help understand Shigella is that the bacteria harbor about a 220 kilobase virulence plasmid that is separate from the bacterial chromosome. This plasmid is very important for encoding things like the invasion proteins, other proteins that are important for infection, as well as the type 3 secretion system. And the type 3 secretion system is composed of many proteins and is an efficient way to basically allow bacterial proteins to be injected through the needle and directly into the host cell cytoplasm. So this process really facilitates invasion and the intracellular lifestyle in the colon cells. Now, one host factor that's been known for quite some time that Shigella utilizes is temperature regulation of the virulence plasmid. So at environmental temperatures or room temperatures like 30 degrees Celsius, the expression of the virulence plasmid is turned off. However, at 37 degrees Celsius or in body temperature, the expression of the virulence plasmid is turned on. So as I mentioned, I've worked with Shigella for quite some time now. And one of the things that always interested me was how does the bacteria regulate gene expression during host transit? And how does the bacteria essentially prepare for infection? And this is important for pathogens like Shigella because they do not want to waste energy expressing virulence factors too early. So we knew, especially with the temperature studies, that Shigella has this ability to really turn expression of virulence on and off at the right time. So prior to a lot of the work we've done, especially looking at small intestinal transit and bile, is that we knew that fecal oral transmission occurred with Shigella, in which as little as 100 organisms can cause infection, and that then we also knew that the bacteria was invading in the colon, where they would find those infrequent M cells, but also need to adjust to environments like the anaerobic conditions in the lumen of the colon, and then overcoming things like mucus and the commensal population. However, there wasn't really much known in between. So if you think about the gastrointestinal tract of humans, there's a lot of additional barriers that the bacteria need to overcome. And that includes the low pH of the stomach and transit through the small intestine. And why is that so interesting? It's because that's the area where this bacteria spends the majority of the time in the human body prior to infecting in the colon. So it's approximately 12 to 24 hours prior to infection it takes to transit the small intestine. And during that time, the bacteria is encountering some harsh conditions, primarily through exposure to bile. And bile is primarily composed of bile salts. It aids in your digestion, but it's bactericidal and is meant to kill bacteria. However, enterics like Shigella and even other pathogens like E. coli and Salmonella are known to be able to resist the bactericidal conditions of bile. And what else is interesting about small intestinal transit and bile is that 95% of bile remains in the small intestine, with only about 5% entering the colon. So at the end of the small intestine and the terminal ileum, you have bile being reabsorbed into circulation, so it is deposited back in the gallbladder. So the overall hypothesis for our laboratory was that Shigella resists bile and uses small intestinal conditions as a host localizational signal to optimize virulence. And so previous work before we got started looked a little bit about the effects of bile on invasion and adherence, primarily through Shelley Payne and Wendy Picking's lab groups, especially in the mid to late 90s. However, no one really looked at how does the bacteria resist bile. So one of the first things we did to get started was look at the physiological range of bile salts. And as I mentioned, bile salt is the principal component of bile. There are other factors that are included. However, for our purposes, we utilized bile salts by adding it to laboratory media and performing our analyses. And in the lab, we use a mixture of cholate and deoxycholate, which is a primary and secondary bile acid or bile salt at neutral pH. And we thought this was a good mix to account for the different types of bile salts that the bacteria encounter during small intestinal transit. In case you're unfamiliar with alterations to bile, the commensal population is responsible for taking primary bile salts and adapting them to secondary bile salts, more so in the colon than in the small intestine. So what we did was we looked at the physiological range of bile salts, which is approximately 0.2 to 2% weight per volume, depending on a person's diet and the actual individual itself. And for the most part, here plotting the average optical density readings, you can see that Shigella was able to resist the physiological range of bile salts, but then that we had slow or no growth outside the physiological range at 5 and 10%. Now we went ahead and confirmed these with looking at bacterial titers to verify the optical density readings. And we were pretty confident to state that Shigella does in fact resist this physiological range. So being a genetics lab, we wanted to know what else bile was doing. And so a great way to do that is to perform transcriptome analyses through RNA sequencing. So our collaborator, Dr. Dave Rasco at the University of Maryland, helped really facilitate many of these studies. And so we were able to look at 3 hours of exposure of bile salts and perform RNA sequencing analysis. We also accounted for different growth conditions such as shaking and static growth, because we were trying to consider what sort of movement the bacteria would encounter as it transits through the small intestine. So we wanted to make sure we were covering sort of both bases in that regard. So bile is pretty harsh condition for the bacteria. So we were able to only limit this analysis to early bile salts exposure, since it was a little bit difficult to isolate RNA at later time points. But as you can see here, despite the growth conditions, we had a large number of genes that were induced in the presence of bile salts that range from different broad categories. So what we can do here is look at interesting gene candidates and test for our mechanisms and our phenotypes of interest, which I will go through some examples today. Another technique we used, and that especially facilitated looking at more longer or overnight exposure to bile salts to sort of consider that 12 to 24-hour time point the bacteria would be spending in the small intestine, was to utilize a transposon mutant library. So we utilized the EZTN5 system to create a nice mutant library. And then we would replica plate onto bile salts and look for mutants unable to grow at low or high concentrations of bile salts. So we have some significant number of gene candidates to pursue. And so one example that I'll show here is the bile salt resistance genes that we've identified. So we found that the ACRA and ACRB genes were induced about fourfold each in bile salt exposure. These genes encode part of the ACRAB efflux pump that is very important for antibiotic resistance. So in order to confirm the importance of these genes, we constructed an ACRB mutant. And as you can see here, the mutant was unable to grow in bile salts. And then once we complemented the mutant, we were able to restore growth. So we were pretty confident at this point that this is a strategy by which Shigella resists bile salts. And we are currently pursuing additional resistance mechanisms. And I'd just like to note the importance of the ACRAB efflux pump. It's really important for enabling resistance to many antibiotics for bacteria, where the pump will basically expel the compounds out of the bacterial cell. And it also ends up being a very conserved mechanism to resist bile salts for multiple enteric pathogens, including E. coli, Salmonella, even Vibrio. So the sheer fact that enteric pathogens are exposed to bile is almost priming them for a reduced antibiotic sensitivity or increased antibiotic resistance, depending on how you want to look at, simply by utilizing the ACRAB efflux pump. So we think this has important implications for future therapeutic development, especially in the age of antibiotic resistance. So one thing we then noticed as we were growing the bacteria, especially as we got to longer time points in bile salt exposure and then overnight cultures, was that we found that there's this white precipitation and clumping phenotype that was occurring. And it happened in both shaking conditions and in static growth conditions, where we would see this white precipitation and an adhesive ring in the shaking tube, or we would see white precipitation settling to the bottom of the wells, and again, this clumping phenotype of the bacteria. It was interesting to note that we noticed this phenotype when we grew the bacteria in tryptic soy broth with bile salts, but we did not notice this phenotype when we grew the bacteria in LB and bile salts. And so the difference between the two media types is the presence of added glucose in TSB. So this clumping phenotype really started to look like a biofilm formation to us. And in case anyone's not familiar with biofilms, it's basically a protective niche to help bacteria resist harsh environmental conditions or even antibiotics, where it's communities of bacteria basically embedded in this matrix known as EPS. And so what happens is you have bacteria attaching to the surface and then starting to produce the EPS matrix in the microcolony formation, and then maturation and stabilization of the biofilm. And usually at some point, there's a signal that enables the bacteria to disperse the biofilm and continue leaving a planktonic cell state. So one of the best ways to quantify biofilm formation is through crystal violet staining, where you basically just add this blue dye to quantify the biofilm formation. So we did this with overnight cultures for Shigella flexneri, 2 different strains, and then as well as Shigella dysenteriae and Shigella sonnei. And so what we're plotting here is, oops, sorry about that. In media without bile salts, we basically did not detect any significant biofilm formation. However, media with bile salts across all the different species, we were able to detect robust biofilm formation. We also performed scanning electron microscopy to really look at the biofilms and the clumping of the bacteria that you can see here. And then most importantly, we utilized fluorescence confocal microscopy with a FITC labeled antibody to detect the EPS matrix. So as you can see here, bacteria grown without bile salts had minimal expression of the EPS matrix. However, bacteria grown overnight in bile salts had robust expression of EPS. And we were actually able to quantify the matrix production with a fluorescence plate reader. And this also allowed us to look at the contribution of glucose in biofilm formation. So what we're plotting here is the fluorescence readings on the y-axis. In light green is bacteria grown in LB media. In dark green, it's bacteria grown in LB plus bile salts. And then across the x-axis here is different concentrations of glucose. So as you can see, when no glucose is present, despite having bile salts in the media, we do not detect significant EPS production. However, as soon as we detect or add glucose to the media, we can detect significant expression of the EPS matrix at various concentrations. We do control for differences in bacterial replication. And basically, we see this really nice on-off switch once glucose is considered in the biofilm formation phenotype. So both bile salts and glucose are important. And we basically want to point out, though, that despite the biofilm formation being so prevalent in the TSB media or LB plus glucose media, the bile salt resistance phenotype doesn't necessarily require glucose. So this graph here on the left is the first graph I showed where we conducted the resistance analyses in LB media with bile salts. The only thing I did here was change the y-axis to match what we need to utilize for when we include glucose in these types of growth analyses. And as you can see here, when glucose is present, right around 3 hours or 4 hours, depending on the bile salt concentration, we see a significant increase in the optical density readings. It does not correlate with bacterial growth since we are able to verify that through plating and determining the bacterial titers. However, we can actually see in the lab the white precipitation that forms, and we actually call this the pop in the lab. So we can see the biofilm formation as it happens in real time. And so finally, I will present a model of what we think is happening as the bacteria transit through the small intestine. But first, I wanted to account for dispersion because I knew that we would have to account for the fact that if Shigella is invading in the colon and forming a biofilm in the small intestine, how and when can the bacteria disperse the biofilm to cause infection? And so here's where I'd like to remind you that 95% of bile remains in the small intestine. And then also, as you digest food, glucose that becomes available as you begin to break down food in the upper small intestine is then depleted as you get down toward the lower small intestine. So we were interested in knowing what signals could potentially lead to a dispersion analysis. And so what we did was grow the biofilms overnight in glucose and bile salts, and then resuspended on the next day in fresh media with basically PBS and with or without glucose, and then PBS also with or without bile salts. And then what we did was quantify the release of the bacteria into that media. And so what we're plotting here is the dispersion and the amount of bacteria that are dispersed into that media. And as you can clearly see, the presence of glucose did not matter in dispersion. It was the presence of bile salts. So without bile salts, we detected a significant dispersion event. And then if bile salts remained in the media, we did not detect significant dispersion. So A model that we are proposing for small intestinal transit of Shigella is that in the upper small intestine or in the duodenum, Shigella first encounters bile salts and is able to resist that initial onslaught of bile salt. Then as food starts to become digested, the sugars become available. Then in the main portion of the small intestine in the jejunum, Shigella is able to form a biofilm and traverse down the jejunum. This helps explain why the bacteria likely might not be able to make contact with the epithelial cells if they're embedded in this biofilm as they traverse down the small intestine. During this time, as digestion proceeds, sugar availability decreases. And then in the terminal ileum is where we have minimal sugar availability and then significant reduction of bile salts as it's being reabsorbed into circulation. And so that we feel is a signal for the bacteria to disperse the biofilm and enter the colon. And here we're proposing that the bacteria are sort of hypervirulent as they're released into the colon for a few reasons. As I mentioned before, previous research really showed an induced adherence and invasion phenotype with bacteria exposed to bile salts. We were able to confirm a lot of the adherence work. And then also the induced virulence gene expression we detected in our RNA-seq analysis pointed to this sort of hypervirulence phenotype growing the bacteria in these conditions. So we were really interested in adherence because adherence is important for both biofilm formation and contact to the epithelial cell surface. So we did a basic experiment where we looked at adherence to the cancer cell line HT29s. So when we grow the bacteria in regular LB media, and then we add them to the infection assay, we get limited adherence. It's not great. However, if we subculture the bacteria in glucose and bile salts and then apply them to the infection assay, we get a significant induction in adherence. And I'd like to note here that we are trying to mimic the small intestinal transit and entry into the colon. So we will subculture the bacteria in these conditions, wash them with PBS, and then resuspend them in tissue culture media prior to applying them to the epithelial cells. So please note that glucose and bile salts are not coming in contact with the epithelial cells themselves. So we had a really nice adherence phenotype. And so we asked the question, does Shigella make adherence factors? And this is actually an important question for Shigella pathogenesis research. In my postdoc, I previously identified two virulence proteins, the type 3 secretion effector proteins, OSPI1 and E2, that are 99% identical. Normally, these are secreted through the type 3 secretion system and help maintain the integrity of the epithelial cell during invasion and intracellular replication. However, we found that once the bacteria are exposed to bile salts, the genes are induced. And the study I performed during my postdoctoral work was we found that the proteins are then secreted through the type 3 secretion system, stick to the outer surface of the bacteria, and are there to function as adhesins. So that was the first time we were able actually to identify adherence proteins associated with this phenotype in Shigella. However, whenever I analyzed a mutant of OSPI1 and E2, I always had this background level of adherence. So we really wanted to ask the question, does Shigella make traditional adherence factors like you would see in E. coli or Salmonella? And again, this is an interesting question because the literature consensus at the time was that Shigella does not make adherence factors. And that was due to two primary reasons. The first is that when you grow the bacteria in laboratory media, you don't see adherence factors. They're very bald, as you can see here. However, if you grow other bacteria like E. coli in the same conditions, you can very clearly see adherence factor expression. Coupled with that was that many of the genes responsible for encoding the adherence factors were annotated as pseudogenes, and many people think that the genes were not actually present in some strains of Shigella. So those two observations together led the field to conclude that Shigella doesn't make traditional adherence factors and didn't think they were needed because of the invasion phenotype. So first, before we wanted to look at the genetics of the adherence factors and possible genes. We wanted to confirm that we could in fact see them in our conditions. So at this time, we wanted to look at both the biofilm assay and infection. And we were able to segue away from the HT29 cancer cell line and utilize an organoid infection model that's become very popular in the field and has been independently developed here at the center by Dr. Stefania Senger. So in case anyone's not familiar with organoids, it's basically a system that allows a more physiological relevant model for infection analyses or any disease of interest actually. And you can actually propagate organoids in the lab by isolating the crypts and the stem cells in the crypts of the intestinal lumen, and you can propagate them in culture where they will form these spheres or the spheroids. So you can do a lot of infection analyses with these or any other type of analyses that you're interested in. For a bacterial pathogenesis standpoint, it's difficult to use spheroid cultures because the apical surface is on the inside. So if you want to apply bacteria directly to the apical surface, it becomes difficult. Well, at the center, they were able to basically develop a monolayer model since you are able to trypsinize these spheroids and seed them onto a transwell system. And then with other differentiation reagents in the media, you can get a more physiological relevant model in that we can produce enterocytes, mucus producing goblet cells, and even the antigen sampling M cells. So here's just a top-down view of scanning electron microscopy of the monolayer model where we get nice cell-to-cell contact. Anything that is lighter like this are the enterocytes that express the microvilli. Anything darker are cells that do not express microvilli, which are typically the M cells. And we went ahead and verified that we do get robust expression of M cells with our media conditions. And here's just a nice transmission electron microscopy images of an enterocyte where you can see the microvilli and the tight junctions being formed. And you can also do things like confocal immunofluorescence analysis. Here we're staining for mucus to show that this model produces a significant amount of mucus and again is a nice physiological relevant intestinal model to use in the lab. And so we have characterized Shigella infection on the model. And so here I'm just showing a nice scanning electron microscopy where we do detect adherence to enterocytes with microvilli. And then we also see evidence of the bacteria able to penetrate and transit through the M cells. We have a preprint where we summarize a lot of this work and it goes into significant amount of detail. But basically, to briefly summarize it, we can apply the bacteria to the apical surface, detect M cell transit. We get the best rates of invasion when M cells are present. So that indicates they are transiting the M cells and invading at the basolateral pole. And we also get the best invasion rates when we use models derived from the colon as opposed to the small intestine, which suggests tissue tropism for the bacteria and helps answer the question again, why is Shigella targeting the colon versus the small intestine? So we're able to utilize this for our adherence factor analyses. So here I'm showing that if you grow the bacteria again in our subculture conditions of bile salts and glucose, and then you isolate bacteria from the biofilm, we see really nice expression of adherence factors now in these conditions. And then we can also see the adherence factors during adherence to the apical surface of the organoid model. And so doing these sort of analyses, especially in the biofilm, we could really see three different types of structures. They were thicker structures, as you can see here on the left, thinner structures here in the center. And then we did detect these protein aggregates very often, and we did a lot of controls, especially for the reviewers, to ensure that we were not getting cell lysis and that these were in fact protein secretion. We detected this not only in our strain that we are using a Shigella flexneri, but as well as clinical isolates of Shigella flexneri to indicate that it wasn't some weird fluke with the strain we were working with. And then looking at the binding again at the epithelial cell surface, the thinner structures really seem to be important for binding to the microvilli on the surface of enterocytes, and then the thicker structures and the protein aggregates were binding nicely to what appear to be M cells. So now genetically, it's an interesting story. If anyone's not familiar with adherence factors, there's different types and many genes and components are involved. And to just very briefly summarize that, you have many structural subunits that are organized together to make the structure, and then you can have some minor subunits depending on the type of adherence factor you're looking at. And then usually there's some sort of pore or usher that allows secretion of the structures to be secreted and organized on the outer surface of the bacteria. So type 1 fimbriae is very common, and there's also curli, which involves different types of genes. And curli are interesting because they form this nice little curled-like structure, which is how they've gotten their names. So many genes are involved. Now for Shigella, based on the appearance, it really looked like the thicker structures were long polar fimbriae, the thinner structures were type 1 fimbriae, and then the interesting protein aggregates really looked like curli to us, even though they were disorganized. So genetically, the genes were present for all three structures, and then we were able to take our RNA sequencing data and map them and saw that these genes were in fact being transcribed. So genes in blue represent structural genes. Genes in green represent the chaperones that help facilitate secretion of the individual subunits. And then genes in orange are mostly the pores or the usher that allow that secretion. Anything checkered or lighter looking, these genes were annotated as pseudogenes. So as you can see an example here, we have nice expression of the structural subunit. And then despite this pseudogene annotation, we were able to detect transcription. We went ahead with different primer sets to confirm the results of the RNA-seq results. So we could either amplify individual genes or even larger segments and operons. So we went ahead and did that for the LPF, the type 1 fimbriae and the curli. So the type 1 fimbriae were again annotated to be pseudogenes or not to be made, was primarily due to the FimD gene encoding the pore. There's a transposon insertion relative to E. coli here at the 5' end of the gene. And so this gene was annotated as a pseudogene. However, as you can see from our RNA-seq results and our RT-PCR results, we get about 90% expression of the gene. And then if we did some analyses where we looked at primers that spanned the upstream and downstream regions of the transposons, we were unable to amplify anything in that regard. So we do think the transposon insertion is accurate for the annotations, but despite that, we still get significant expression of the genes and the operon. And then for curli, it's an interesting story because the structural subunit CSGA was also annotated as a pseudogene due to transposon insertions as well. But again, as you can see, we get nice expression, about 90% of the gene relative to E. coli. And then also the pore for CSGG is actually truncated in Shigella, and we were able to verify that with our analyses. So most of the genes are intact, they're being transcribed, which is really good indication that the bacteria are in fact using them and expressing them, especially pathogens like Shigella that doesn't want to waste energy making something it doesn't need. We also detected additional homologs that are present that can compensate potentially for any pseudogenes if needed. So one last thing we did was to confirm that, yes, this transcription is real, was to look at induced transcription levels through quantitative RT-PCR analyses. So we looked at the literature and found that roughly many of the structural genes can be induced by glucose. And so when you control for differences in growth due to adding glucose to the media, see here with our quantitative analyses, all the structural genes were induced in the presence of glucose, especially LPFA, FimA, and CSGA. We have our control gene here that is in which the expression is not induced in the presence of glucose, as to make sure we weren't doing anything wrong with our analyses. So we're really confident at this point that these structural genes and other genes are in fact transcribed for Shigella. And since we're a genetics lab for the most part, we went ahead and made mutations in LPFA, FIMA, and CSGA. And as you can see here, the mutants had reduced ability to form a biofilm and also reduced adherence to the organoid model. And then a genetics lab will complement the mutations to make sure we can restore the phenotype. So we did so for each of the 3 candidates. And while doing so, we also added a histidine tag onto the 3' end of the gene that will help facilitate future protein analyses with these genes and proteins. So we transformed them back into the respective mutants and performed TEM and analysis and biofilm formation. Now, just as an example, I won't show all 3, but it was very consistent for all 3. You can see here the FIMA mutant loses those thin structures. However, we're able to restore those thin structures back with the complement. And the complements were really nice because it really helped to verify the appearance of the structures as well. And then nicely, the complements, even with the histidine tag, are functional. So we were able to restore biofilm formation relative to the same levels as wild type and significantly higher than the FIMA mutant. So we're really confident at this point that we've identified 3 structures that are expressed in these conditions: the long polar fimbriae that are thicker, the type 1 fimbriae that are thinner, and then the curli. Despite the disorganization that we're detecting in Shigella, they are functional from our analyses. So we're really excited to move forward with this work, and we're hoping to have some exciting data about some more information about the proteins and secretion of these factors as we move forward. So with that, I'd like to conclude. I hope I convinced everyone that we feel like we've really expanded the Shigella infection paradigm to really account for transit through the small intestine prior to infection in the colon. So Shigella is able to resist bile salts. It's able to form a biofilm to help facilitate small intestinal transit. During these conditions, there's extensive gene expression changes that are happening that help prime the bacteria for an anaerobic environment in the colon and also help the bacteria to prepare for actual infection in the colon. And so once we can account for things like dispersion of the biofilm, we think a nice hypervirulent form of the bacteria is entering the colon for infection. And so we really think as we pursue a lot of this work into the future, we will be able to identify novel therapeutic targets and also considering all the changes that occur to the bacteria as it transits the host so we can hopefully develop those more effective vaccines. So with that, I'd like to thank you again for this ability to present today and happy to take any questions. I first like to acknowledge members of my lab and collaborators, both past and present. We've had a lot of people, a lot of students work through our lab that have taken pieces of everything and moved it forward. I'm very much grateful for all the laboratory interns we had. Most of this work presented today was facilitated by the postdoctoral fellow Courtney Nickerson and a former student and technician, Rachel Shannon, who is now at UT Southwestern and is about to defend her thesis. So I'm very excited for her. And we have had other members of the lab who've helped with this work and now post-COVID shutdowns have helped us open up and get going again. Our collaborators at the center, especially Dr. Senger, Fasano, and Fiorentino for utilization of the organoid model, Dr. Rasco for the RNA sequencing analysis at Maryland, and then Ms. Ann Tidsdale and Dr. Kumar for assistance with the scanning and transmission electron microscopy. And our funding is primarily through the NIH and the Bill and Melinda Gates Foundation. So thank you very much.
Great. Thank you so much, Christina. This was a really exciting talk about an important pathogen, and you sounded almost apologetic for being a genetics lab. But thank you so much for making this so clear and understandable for someone who does not come from a genetics lab. So anyone is free to type in questions. I have a ton of questions, but I will, of course, give priority to those listening and typing in questions. The first comes from Yersinia 43. Sorry, it's a fairly anonymous name, but clearly a pathogen, a nerd just like us. Are people who have had their gallbladders removed less likely to get shigellosis?
I've wanted to answer that question. So that is something interesting to consider. I do feel like there's probably other signals they're utilizing to help prepare for infection, especially as they enter the anaerobic environment for, infection in the colon. And keep in mind that, Shigella grown in laboratory LB media can cause infection. It just does it so much better in the conditions that we're replicating here. So I do think there might be an impact on that. And I've been curious to see if there's any correlation between the two, less infection or reduced susceptibility. Yeah.
Cool. Great question. Next one is from Joy McKenna. So thank you for a wonderful talk. I'm curious about the Shigella resistance to bile salts phenotype. How does this relate to the upregulation of type 3 secretion system genes in the presence of deoxycholate?
Right. So interestingly, with the mix we were using, a lot of the type 3 secretion system genes that we found that were induced were not necessarily genes for making the type 3 secretion system needle or for invasion, but that helped facilitate later on an infection. So we are starting to look at those and see what that means. And the best example is OSP-E1 and E2 for the adherence mechanism. We do see induced secretion of the type 3 secretion system in these conditions as well. So it is interesting to sort of put everything together with having enough energy to resist bile and then also regulate virulence at the same time. And we're hoping to answer some of those questions more so in the future. Interestingly, with the ACR-AB, the other component, the outer membrane component of that pump, TOL-C, wasn't significantly affected. So we think there's some mechanisms involved with that could be interesting to pursue. But clearly we're able to see the contribution of the pump and other changes that facilitate bile salt resistance, and at the same time see this induced virulence phenotype.
Great, I hope that answered your question, Joy. Next one, are there other pseudogenes that don't produce functional proteins but are still transcribed in your RNA-seq studies?
So we might be looking at a few candidates, but so far everything that we have seen that is least transcribed, we have the evidence, at least through mutations, that the protein's likely being made. And I maybe was a little bit too apologetic about being a bacterial genetics lab, because we do need to do a lot of the protein verification work that we're hoping to do with this and ensure that the translation step is happening. But so far, it looks like anything that's transcribed is in fact being made into proteins. Yeah.
That was really cool. I also thought the transposon insertions into these structural genes for the attachment mechanisms, which are clearly very important. Actually, I'm going to butt in here because I had a question about the attachment mechanisms, and you identified beautifully the 3 different mechanisms that were all attaching to different structures in the epithelium. Are all 3 necessary, do you think? Absolutely necessary?
I do. Yeah, sorry. Yeah, I do think so. I do think there's differences. So it really looks like FIM-A is binding to, it seems to be the most important for adherence and binding to the enterocytes. But then also it looks like the long polar fimbriae and the curli seem to facilitate binding to maybe the M cell surface and to initiate that contact as well. And then also there's the idea of, of compensation. So if one isn't expressed properly, that they'll still be able to produce something that helps facilitate adherence. So yeah, we're really excited to keep pursuing that and go in that direction.
Cool. Lots of questions here. Question from Kate Baker. Thanks, that was great. I wondered whether you saw any action in the flagella pseudogenes in your studies.
To be determined, but yes, we're interested in pursuing that as well. we have some interesting changes that we hope to pursue. Yeah.
So keep an eye out for that.
Keep
Keep an eye out for updates. Next question is from Satvik Vupalati. Thank you for the great presentation. In terms of transmission, I'm curious as to if Shigella can be transmitted sexually. So there tends to be a high correlation of infection with HIV patients.
Yeah, so we think that has a lot to do, especially with Shigella sonnei, seems to be more prevalent with that, which is very interesting. And actually, Kate's work looks into that very extensively. So definitely, I would point you to her studies. But it feels like there could be basically as an STD sort of a contribution to the fecal oral transmission, and it's very prevalent with HIV, which is interesting. So yeah, it's interesting to think about potential implications of HIV infection and Shigella spread, especially when it comes to, things like T-cell immunity and things like that. So.
Okay, we've just got a couple of final comments. Thank you, very interesting. And Kate Baker says, let's talk, smiley face. So you'll be contacted, I guess. I have one final question from coming from the world of beneficial host microbe interactions. Are these mechanisms also used by beneficial gut microbes, do you know, to survive passage through the stomach and survive and persist and thrive within the gut?
Right. So we did actually start looking a little bit at the escape pathogens, or Enterococcus, Staph, Klebsiella, Acinetobacter, Pseudomonas, and Enterobacter. And Enterococcus, which is sort of that commensal / pathogen, curiosity state of that bacteria. So a lot of these pathogens as well, or bacteria, utilize ACR-AB and also are changing during bile salts exposure and changing other aspects of their outer membrane to be able to resist bile and then colonize the intestines. And actually some research really needs to be done with that. So it seems like if bacteria are going to at some point transit, even if it's a commensal, it needs to resist bile, it needs to get through that and then establish colonization in the colon, where you'd then be more exposed to the secondary bile acids and things like that are being altered by the commensal bacteria themselves. So yeah, there's definitely something there to it. And especially with the escape pathogens, where it feels like they're not really causing infection in the colon, but will maybe cause infection at other sites of the body. It does seem like they might be transiting through and have a phenotype there that could affect then antibiotic resistance as well. So, yeah.
Okay, then we'll wrap it up. And thank you again, Christina, for the great talk today. Very interesting.
Great. Thank you very much again for having me, and happy to talk to anyone who would like to talk that was listening in. So.
Yep. So thanks also to the audience. Tune in again next time for the next microseminar. Contact Christina if you've got any questions. And have a nice afternoon or evening or morning, whatever it is. Okay. Thanks a lot. Bye, everybody. Okay. I think I've ended the